I am very excited to introduce Dr. Sandra Loesgen from University of Florida Whitney Laboratory.
Go Gators!
Yeah.
Champions!
Yay. She's in St. Augustine, Florida, so not in Gainesville. Anyhow, Sandra actually comes to the US from Germany. She did her PhD and her first postdoc at Georg August University in Göttingen. Good, okay, I got the pronunciation right. And then after that, she came here to do a postdoc with William Fennekel, a very famous natural products chemist at the Scripps Institute in San Diego. She spent a little time at Janella Farm, which is part of the Howard Hughes Medical Institute, looking at brain-active natural compounds. And then she was at the NIH and IDDK to do another training on HIV inhibitors. And all of this is with natural products from different organisms. She started her faculty career at Oregon State University. And from there, she moved on to University of Florida, the Whitney Laboratory for Marine Biosciences. She has more awards than I can go through in a short amount of time, many papers, NSF, NIH grants, and she's a fantastic collaborator. We met at the pharmacognosy meeting a couple of years ago. I was peddling our anti-parasitic screens, and she had a compound, so, and the rest was a wonderful collaboration. So I give you Sandra, and I think you will love her talk.
Thank you. Well, all right. Yeah, thank you, Rob, Kevin, everybody here for having me. It's my first time in Minnesota. I can't believe it. So very excited to be here. It's a little cold. We had 30 degrees C when I left. It's warmer now. And I think I freaked some people out. I took photos of a snow pile next to my hotel room, so next to my hotel. Okay, let's see if I can move the slides. Kevin was better than me. What did you do special that I don't do? Huh. Now it works. All right. Okay. Thank you so much. Sorry about that. All right. So I want to talk today about the work my laboratory has been doing now for over 12, 15 years almost, starting in Oregon State, the Beavers, and then going to the University of Florida, the Gators. And what we are working with are actually microbial organisms. So we work with fungi and bacteria. I show some here. And I always love saying jokingly that I am the chemist head in the lab, but the real chemists in my laboratory are actually all the little microbes and their beautiful chemistry that they are making. And they're providing us tools to study medicine, to study diseases, pathways, cellular biology, right? So I want to take you a little bit today on a little journey about several microbes that we have studied and their application that might be of interest to you. I want to give a little background. So I was looking up where I'm going. I was like, wow, this is the most north I've ever been. So it was very exciting to come here. And again, my first trip up north. I'm coming from the University of Florida. The mothership is in Gainesville, right in the swamp, big university down in Florida. But my institute is a marine institute. So we are actually a satellite campus of the University of Florida. We're right there at the Atlantic coast, so much, much nicer. And if you haven't been to St. Augustine, maybe some of you have been to St. Augustine, Florida, it's the oldest city in the US. There's some really cool public radio and public TV shows on the first Spanish colonies that there were way before Jamestown, way before Williamsburg. So it's really a fantastic old city. So if you come to that area, highly recommend to visit St. Augustine, shown here. So our laboratory, as I said, is right at the beach, which is fun in many ways, a little concerning during storm season. So here's the Atlantic Ocean. Here is the bi-coastal pathway. So here's where all the ships coming through, a lot of sailing boats going down here through to the Bahamas. So it's a beautiful piece of Earth that we're really happy to do our research at. what is new for us, and we're really excited, we get a new laboratory. And we were pushing really hard to get that lifted off the ground. We want to be high up and dry. We don't want to have hurricanes to wipe us out. So the new building is in progress. And just to give you a little show here, we have actually a little time lapse of that process of getting us a new research institute. Hope it plays, let's say. Nope, go back. I have a video here. We should have checked it probably. No, it doesn't play it, I assume. Anyway, I'm sorry about that. It's a little. Oh, yes, you're smart. Come here, mouse. Over here. That is. It's done. So just to give you a little bit of a fun little background, we have this webcam that people can log into online. It's kind of fun. So you see the Atlantic Ocean in the past. You see the bicoastal pathway, the Matanzas rivers on the left. It's very fun. We have like huge tides in that river. So we have an open to the Atlantic Ocean. So we have huge tides coming in and out of that river. So lots of students that go kayaking there actually have to paddle really hard to come back to the laboratory, and it's a little risky. Haven't lost one yet. But it's a beautiful new campus that we are building there. It's a small campus, only 12 faculty, around 70 researchers on site, but we're really happy. And you see, I was hoping to get the building higher, but it is in a good elevation for now. Sorry, do you need me? Okay. All right, so what I want to do today is to make this fun for you guys is I want to take you to a little journey around the globe. Everybody likes to travel. And I will just tell you some stories about microorganisms that we have found all over the globe, usually in collaboration or under permits that we can sample there. And we brought them back to us, we studied the chemistry, and then we found an application for these molecules. So I want to show you in different little stories, what have we found in terms of the microbe, what is the chemistry that we found, and the most interesting probably for you guys in the room, how can we use this chemistry to study diseases and cellular pathways? Do I do something right or wrong?
No, you're fine.
Okay.
Sure.
All right. So a little bit of background, and probably most of you guys know this in the room, but natural products, molecules that are made by plants, microbes, fungi you find, they have huge potentials to be drugs, right? And when I started at William Fanical's lab in San Diego, studying for natural products, he always showed the slide. It's actually his slide, and I'm glad to use it now, because what it shows are these two gentlemen here studying microbes on agar plates and those slant dishes here. And most students, when I ask them, know who's the gentleman on the right. Anybody here in the room knows who that is? That's Alexander Fleming, right? So Alexander Fleming found penicillin, very potent antibiotic, tens and more generations in the clinic, obviously, made by a fungus to prevent bacterial growth, right? It's a very used applicable structure. But then also on the left here is Salman Waxman. And most people don't recognize this name at all, but he studied his whole career at Rutgers, and he found penstrep. If you do cell culture work, most of us probably do in the room, penicillin, streptomycin, this combination. The streptomycin is coming from bacteria that he's been studying his whole career. One of the first tuberculosis drugs come out of his laboratory at Rutgers, right? And so here are the structures. Many of you might not be as excited for them as I am, but as an organic chemist, I look at them and I get excited, right? Look at these complexity, they're different header atoms, different rings, different side chains, sugars. This is something that nature doesn't do lightly. This is programmed and evolved to do something for these microbes, and we have to figure out what it is, right? And that's kind of the task. And again, these are good drugs. 50% is the lowest estimate on how these molecules actually inspire and developed into the clinic. FDA-approved drugs, right? In infectious diseases, this number is up to 75%. So compounds found in nature, and that's plants, bacteria, fungi, you name it, even animals, can make it into the clinic, can make good drugs. And that's what motivates us. So how does it work in my lab? We are basically brewing some kind of beer here. We're taking our microbes, we culture them, sometimes big, sometimes small. We kind of look what's there, we concentrate the organic essence, and then we try some simple activity assays. And I say simple because that's probably, compared to my colleagues in the room who are much more esteemed, we're trying things. We're trying to kill cancer cells. We're trying to find HIV drugs, but we try to find initial activity. So maybe I would say our team is evolving this part in natural product-based research. We try to find smart assays. Which of the microbes that we get from all over the globe, which one is worth our time, our effort to look for chemical entities? And we've done some interesting things, from having assays that are protein-based, just binding to a protein in HIV, having an assay, obviously the classics, cancer cells, antibiotic testing, gram-positive, gram-negative, a huge need, obviously, to find new antibiotics, but also going into whole animal models. What does this microbe do in chemistry purposes to affect the development and maybe the diseases we can cause now in those models like zebrafish? Down here is a part that I don't want to bother you with, but this is the part where the chemist comes out. How many molecules do you think does one microbe make? Usually not one, it's usually more, right? And so up to 1,000 chemical entities we get of this juice from these bacteria and microbes, and that's a challenge. So down here, that's what my students in the chemistry program learn, is how to purify this molecule that is bioactive, how to figure out their structure, how to figure out their three-dimensionality, because no other company will pick up our drugs if they are not fully assigned, right? And let me know if there are questions or anything. Happy to answer. Okay, so a little bit of stuff we did last year. So I just want to show you a few structures to just ease you in. One study we did last year was on these halogenated compounds. The perks being in a marine institute is you find microbes that can use bromide, chloride, all this stuff in the ocean. That's really fun. So we found these molecules, and in this collaboration with April Reisinger, San Antonio Health Center, we found that these really interesting molecules look a little bit like a propeller, but they work really nicely against triple-negative breast cancers. As you know, cancers that are hard to treat, not many receptors to bring stuff in. And as a project we were pulled into, once you arrive in Florida, everybody wants to work with manatees, right? They're the choppy little, I don't know what to call them, cows? They're the marine mammals, protected, and they die every year now. The seagrass they're eating is in decline, huge problems. And a colleague of us at UF started saying, hey, we got to look for the seagrass. There's something that is devastating the seagrass in our bays and our mangroves. What can we do? And working with them, we looked into how are the microbes there making chemistry to prevent infection against pathogens of seagrass. So that was a fun story. We also look into genetics. Like, we found molecules like this in the past. Phenylactones is a bacterial metabolite. And then we often collaborate with other enzymologists. How is this actually made? How does nature put together these complex structures? But these are all the things from the past. What I want to do today is a little more fun. It's like a journey around the globe. So I think we have 3 stops today, if I make it on time, to land you. So let's go for a little journey. First one, Ohio. I've never been, but I have a collaborator there. I got to get an invite. Don't say that. Well, so one thing we started is looking in insect-associated fungi, Basidiomycota. And this all started when going out of your comfort zone. That's what I'm doing right now. I'm talking in a vet school. I'm a chemist. What am I doing here, right? But going to a conference of mycologists. I was invited to go to a mycology meeting, and I do the chemistry of fungi, but I have no idea of anything else fungi, right? So I was approached by this colleague, Jason Slott here, and he says, well, we have this weird fungus that lives in termite nests, and it looks like the termite eggs. So people call it the cuckoo fungus, right? The idea, I put my egg in another nest and everybody gets taken care of, and I have a nice life, right? And here's a picture of it, actually. So here are the lower termites, whoopsie, that we are, oopsie, that's kind of preview there. Here are the lower termites. That's their eggs in a nest, and their eggs are actually oval and clear, while the termite makes little balls that are more darker, more brown and orange here. And that's the same phenotype we can see on this fungus on an agar plate. We can take a brush and take off these little pearls, tiny little powder bits of this fungus. And the termite is tricked into keeping this fungus here. They actually lick the eggs, pseudo-eggs, the fungal balls. They lick eggs and fungus. They pile them, they reorganize them, they treat them as their own. Interesting, right? But Jason is actually not really interested in insect and fungal symbioses or pathogenicity. He's actually an expert worldwide for magic mushrooms. So psilocybin, the compound that you might have heard of that is in magic mushrooms, like the macrofungi that you can eat for whatever reasons, he found the genes making psilocybin in this fungus. 4 of the 5 homologs needed to make psilocybin are in this fungus that's hiding in termite nests. So he approached us saying, I see those genes. Can you tell me psilocybin is made, yes or no? And maybe that keeps those termites in check and happy. So what's happening here, right? Is there a termite party? What's happening, right? It's funny when you type termite party in Google. So that comes out. And so our goal was to find psilocybin from his fungus and see if it's really there. And maybe that's the chemical that makes the termites happy or accepts the fungal cohabitant, right? So we went in here, and I'm sorry, that's of chemistry, but we didn't find psilocybin. Bummer, maybe, but I think we found something better. What we found, and that's all work by a postdoc at the time in the lab, Annika Jagels, she took this fungus and this powder and extracted this molecule here. It might look really simple to you, but this is a novel alkaloid. This skeleton never been seen by man or termite before. So we were stoked, right? Psilocybin looks very simple. This looks simple too, but never been seen before. No literature evidence. You can search the structure, databases, nothing. It was fantastic. And she can also find it if she treats the extraction a little differently. She finds it with a sugar moiety. Sometimes plants do this. They're kind of masking their molecules, and then you can release a compound, like a product kind of idea, right? And so the fungus makes this compound a lot, and it looks like this, and it's new. And we called it, that's fun part of our work. If we find something completely new, we can give it a name. So we called it Athelia pyrrolidine after the fungus and this beautiful 5-membered ring. So that was a fun part. Okay, why are we excited for this? Well, that's just background on how we find it. Well, first of all, you can find it in Ohio. I didn't mention over 70% of termite nests in Ohio, but also in other countries, have this fungus. Nobody knows why, seems to just coexist. And we see this. We can go in the field and find the termite balls, so the fungal structures, and we can see high abundance of our compounds. So that's a good thing having a chemist on hand. They can quantitate things, they can tell you what the structure is, etc. We also see many eggs from the field have this compound, so it might rub off onto them as well. So that's a principle that is really, probably has a function in nature. Next step is, well, it's an alkaloid. Alkaloids are known to do something like psilocybin in your neurosystem, right? They're psychogenic compounds. So we submitted this, and there's a beautiful infrastructure that's called the Psychoactive Drug Screening Program at the NIH, and you ship your compound, and if you're lucky, you get tested against 46 receptors, human receptors. And that's shown here. So on the x-axis, you see all the different kind of human receptors. And then we see binding is made by the radioligand replacement assay, but you see binding to human receptors. And we're really excited to see that, yes, it's active, and it's active against the serotonin receptor. Some people in the room might know psilocybin, the molecule we were chasing, is a very potent 5-HT2A, the serotonin agonist. That's what gives you the kick in the mushroom trips, right? So we're hitting a similar or the same target. Interesting, right? We did some more functional assays, and that's really a screenshot from the email we got. I was so excited, I took a screenshot. So that's what we see here. Our molecule is compared to their standard, which is clozapine. That's a clinically used drug for autism and schizophrenia. And of course, this one is like nanomolar active in their replacement assay. But our fungus molecule here, straight out of fungus, not modified, not optimized, has also 360 nanomolar affinity. So that's jackpot, right? How often does this happen? Very exciting. We followed on with a small synthesis. So it's good to have a chemist in your team, right? So we made this molecule just by a really quick 2-step synthesis. We make this molecule and we compare it to our natural product. Yes, that's the right thing. We're moving forward. And then we test again with a PDSP system. And it's nice because we can see the natural product is here. Our synthetically made compound is here, same kind of activity. And then lastly, phenomenally, they take functional assays. They cannot just say, yes, it binds a receptor. They can also say, is this an agonist or an antagonist on that receptor? And what we found out is it is an antagonist similar to clozapine and doesn't act like serotonin is on this receptor. So we have found a new fungal-derived serotonin antagonist. And that's something I went into patent this year. So we have this patented now and secured for UF. We also figured out, if someone in the room is interested how it's made by nature, we can look into the biosynthesis, how are the components, we can feed that. We proved every single carbon in the molecule, where does it come from? And the side chain, this pyrrolidine molecule, this moiety here, we can show comes from ornithine as amino acid. So that's just proof of principle. We know this molecule is made in the fungus, and we know exactly where the atoms come from. Let's skip that. You might now ask, what does it do for termites, right? And I wish I could answer that. Termites do have serotonin receptors. They are associated with some feeding behavior in termites, which would be good if you think about a patent with regards to an anti-termite compound. But we haven't known the answer yet. So what we're doing is we're working with colleagues also at Ohio State. We try to find that button again. Here we are. And what you see here is one of our first experiments, right? We put our compound, which is not volatile. So it's not an olfactory signal. We put our compound on these filter papers in the middle, and they have to eat them. Similarly, like they would lick and work on these eggs, right? They work in the nest often like that. And we have the different compounds here. And now we have some software to analyze. Do they eat the one more? Do they behave different? Termites are not really good social insects. They don't behave like ants. They're not exploratory. So I don't like them, right? So they're actually a problem. But we have that now on videotape, and we're analyzing that with our colleagues. So stay tuned for the answer. What does this compound do in nature for the termites? Yeah, I always think this one on top, it actually has a compound, is like going crazy, running way more. But that's just me looking at it, right? That's not what we can publish, obviously. Okay, second stop. I'll take you. Yes. Sure. Well, I tell my student not to lick their fingers, but we don't. The next thing would be behavior. There's like a cube. There's a company that offers to test the compound on a mice in a cube, and they have like different parameters, like they show certain videos, they have certain movements and how they behave to it. So there are ways to do this. It's just all very expensive. And we haven't had the chance to get funding for that ever. So, yes? So we made exactly the natural product because it's very simple. We didn't have to simplify it, but we haven't done any changes on it yet.
Can you patent natural products like that? Buy something.
You're good. You're good. You're good. You know more than, yes, indeed, you cannot patent a natural product. You can patent it as application though. And that's what we did. We did a prodrug application. So with the sugar on it, oopsie, the sugar on it is basically cleaved in any kind of cellular context. And so we put in the patent majorly a prodrug, sugar on it or some other leaving group that will release a compound. And the application towards serotonin receptor involved diseases can be patented. Method of use, correct. You know more than me. Exactly. Fluorine, people love a little fluorine here and there. Exactly, yeah. And that's something we're looking into, but now it was more the excitement. The structure itself is kind of boring in many ways, but the activity, the serotonin receptor, that was what our patent office got really excited. And we had talks now with startups in psychedelic development to work with that compound. Good question. Yeah.
Was it from the developed in the lab, or was it from natural material?
Good question. I was probably first. Yes, the question is, when we first isolate the compound, was it from the lab-grown fungus, or was it from a wild-type fungus from a nest? So we found everything in the beginning, as I showed, from an agar plate, lab-grown Athelia fungus, never seen a termite, basically, or termite egg. But then we went, that's what this one slide was about, we went into in the wild, into the field, we got termite balls, the fungal material. It has high, even more than we have in the laboratory. And so we actually have 2 nanograms of compound per termite ball in the laboratory and up to 4 nanograms per compound per termite ball in the wild. So it is in the wild, but good question, they changed in the laboratory, yeah? Good one. Wow, you guys are engaged, I love it. All right, everybody ready for the next trip? Well, I'll take you to my home country now. We're going to Germany. You know, center of Europe, lots of microbial stuff to do there. And this is a story that started with my PhD. So if you think right now you're done with your stuff in the PhD, don't think so. It might take, you might work on these things for many years to come. Okay, so it started all off with this bacterium up here, which is a Streptomyces botrycensis, a soil bacterium. And it was isolated on campus at the University of Göttingen, where I did my PhD. And it was isolated next to the cafeteria. So here's a roof of the chemistry building. In red is a cafeteria. And next to it in like a bed of greens, someone grabbed from the lab a soil and isolated bacteria. Many, many bacteria in soil, as you know. So, but one of them was this one. And in their initial assay, it killed all the cancer cells. And so that was all done previously, but we were lucky to work with this bacteria more and figuring out what does it actually do to work in cancer cells. What's really fun here is, so it got the name, the molecule we found in the end is a carcine, right? It's carcinogenic, it's carcine. But since it was found next to the cafeteria, where are my German speakers? In German, it's Mensa. Mensa. And so we called the compound mensacarcine, the compound from the cafeteria. And it's a beautiful structure, organic chemistry-wise, right? It's a highly decorated a system, lots of oxygens, right? Lots of rings. It's actually really flat. When we crystallize it, it's a rather flat structure. And what's beautiful is it makes a ton. So we can grow this bacterium in 1 liter of medium, and we can crystallize out the compound, 120 mg, up to 200 mg per liter. That's unheard of. Like everything I normally show you guys is like 5 mg maybe, right? So we have a ton of it. That makes studying mode of action. What does it do in the cancer cells? Why cancer cells at all? Much, much easier. Okay, so this is all work done by 2 very talented students. Birte, postdoc at the time. I'm not a cancer biologist, so she brought that knowledge with her, which was great. And a graduate student who learned everything from us, Elizabeth, who's now going down the cancer rabbit hole and actually got a K99R00 to continue that work in cancer. So very proud of her. Okay, so what do we do here? A little bit of background to this molecule. Mensa is a place to eat, right? We studied mode of action. That's a paper. Very proud. Only 3 women doing a powerful job here. Very exciting. So what do? Let's give you the feedback. We work with the National Cancer Institute. Again, those resources are so enabling, right? So we send them the compound and we get feedback for 60 cell lines in their response to our molecule. It was shut down for a while, really devastating for people that look for new molecules, right? And so what you see here is, it's really hard to see, but you have this mean log C50. So lethality, if you see bars going to the left, these cells are dying under that concentration. To the right, they are less affected. And what you want to see, I was told, is more like a tissue selectivity. So here the red block is all the melanoma cells, and they are more susceptible to mensacarcine treatment. So they die easier over the other cell lines. So that's something you want to see for a drug, not a general toxin, all right? And you can see this on these diagrams here. If something is dipping below the lethal concentration here, then we have lethality in the melanoma cells. However, in breast cancer, we have growth inhibition, right? We do inhibit the growth inhibition, or we inhibit the growth of these cells, but we're not reaching lethality. So it does affect cells in general, probably inhibiting their metabolism, but doesn't kill them. And that's what we want to see. So melanoma was a hit. Why melanoma is good? I don't have to pitch that. Maybe I have to pitch it here. So Florida has a lot of melanoma cancers. And one thing that I didn't know before I started this research is melanoma is very prone to cause brain cancers. It's a very highly metastasizing cancer. So there's actually a really cool NPR story of a person that had a melanoma coming into her brain, causing devastating brain cancer, right? So check your skin, be careful if you go out in the sun, right? So it's a very important, it's not the highest rate of cancer death that you can find probably in the US, but it's an important one, right? And so we looked into cellular mechanism of action. That might be something that some of you are familiar with. So we're looking into, here are our melanoma cells. Here we were sensitive and inducing apoptosis. Here's our colon cancer cell line that, yes, is inhibited in growth, but doesn't die on treatment, which was interesting to us. First thing we did is looking for apoptosis. Do we induce apoptosis markers, or do we cleave PARP? Do we look at other paspaces here? And yes, we have full-on apoptotic behavior in melanoma. We don't see this in our colon cancer cell lines. Then we made a fluorescent probe. Always good to have a chemist as a friend. They can take those molecules, they can put a little light bulb on it, and you can follow them in your cells. And what we did here is we put a rhodamine molecule. So here's our mensacarcine, and in one corner, we added our fluorophore, and now we can follow it in cells and see it actually co-localizes in the mitochondria. So it does go in the mitochondria the most and affects cellular metabolism there. That was good. Cellular metabolism also gives rise to metastasis, right? If you have cells that are highly metabolic active, they usually also migrate and move more. And that was a fun collaboration with Amarinder Nain at Virginia Tech. What he is doing is, he is an engineer. He builds these, what he called step systems. So what you see here are actually human cancer cells, and they're using this tight rope to crawl along. That's how cells move, right? They use their bodily activities, if you will, to stretch out, to move on. And so what we were interested in, mensacarcine might stop that migrational movement, the cellular movement. And so really happy that collaborate with him. And what he sees is similar on those videos is, if we end mensacarcine to those cells, they're kind of wiggly. That's normally they would move, right? But with mensacarcine treatments, they stay where they are. They're not moving, and that was quite interesting. And actually, it's similar to if you remove the glucose from the media. Well, cells need glucose to survive and metabolize, but they also need glucose to do the motional, to do the migration. And so having no glucose in the media, he tried that, looks like our mensacarcine treatment. And that's in a mouse myoblast line. Okay, so how does it collect back to our melanoma cells? Well, we were interested in human skin cancer, and here we say, if we can block the glucose uptake, for example, with a 2-deoxyglucose, this is a blocker of all glycolysis in the cell, we see less migration in the myoblast that he is using for mouse. We also see less migration in our melanoma cells. So it has to do with metabolism. It is in the mitochondria. We see problems when we remove glucose with that, but we still wasn't sure of the mode of action. So student Elizabeth, in her second paper from her thesis, then went to town with that molecule more. And what I can show you here is really cool. Vemurafenib is a clinically used drugs for melanoma. And we combined it with mensacarcine. And what came out of that study is synergism. So we get more from these two drugs together than one alone, which was neat. And she could do this in a nice study. So vemurafenib, clinically used drug, is a BRAF kinase inhibitor. You might hear those are actually fastly evolving resistance in the cancer setting. I don't want to upset anybody, but it has to be shown, right? We think we have those BRAF kinase inhibitors and they're gold in the clinic, but they also force us to get quickly resistant. So here's a remission of a patient that has been worked with the RAF inhibition drug, but there is remission really quickly. So these kinase inhibitors, long-term is not a good solution for these kind of cancers, right? And that's Zelboraf, or Zelboraf is the brand, vemurafenib is a chemical that is causing that. We were lucky to get cell lines that were resistant to this clinically used drug. And what you see here is in red, yes, our compound mensacarcine can still affect cell lines that are resistant to the clinically used drug. So the synergism was one idea, but we also might have efficacy when these cells are resistant to the clinically used drug alone. So that was cool. And I apologize, as you see those error bars, they're horrible, right? We had a hard time doing this, and believe me, we did it 100 times. This is due to the cell being so different under the resistance mechanism, being under pressure of this drug all the time, messes up everything, right? So very interesting study here. Question now is, right, metastasis. So how does we look for the migration in that case? And I have this little video here, if it plays, if I find it, yay. So this is now melanoma cells treated with mensacarcine. in Professor Nine's STEP system. And there's a little bit of a pause here, but what you can see is here these protrusions that come out and they grab. They really climb along those ropes and try to move on. Once we do the mensacarsin treatment, what he found is a very unusual phenotype he has never seen before. So these protrusions, he called them, this length of this grabbing arm, are much longer. And that's something he hasn't seen before. And the cool thing is we see this at a dose much lower before killing our melanoma cells, right? So our cancer treatment was at 1 micromolar. Now we are way beyond that. 10 times lower, we see effects on the phenotype of these cells. So the molecule mensacarsin does something to the phenotype, and it might explain these coil widths that they need for migration. This gets smaller with mensacarsin treatment, and that's what they need to migrate. So we might have a compound that is able to stop migration of these melanoma cells, which was very, very powerful. Yeah, so that's metastatic potential. Okay, why is she talking about all that, right? Mensacarsin, old story. Well, it all came to a first initial study about this thing. Anybody knows what that is? Just to wake you back up? Not Rob, she knows it. It's a beautiful picture of a Plasmodium parasites in human cells, right? And we found mensacarsin's new tricks I want to share with you today for the very first time, I think, and Rob agreed. So we sent it out for all kind of testing, right? We knew it does something in metabolism of cancer cells. Melanoma cells are more glycemic, so there's something with metabolism. But then we sent it out to the malaria venture, and they came back with, oh, that's phenomenal. This is killing our Plasmodium parasites at a nanomolar range. Everything in cancer was micromolar range. That's better. And malaria is still a problem. Obviously, we can talk about that. Still in the developed countries, for sure, huge need, especially affects children, as you know. And the pandemic, I thought, oh, we have vaccines now for everything, right? Some vaccines are promising, but the rate from what I was reading about the spreading of malaria, especially with global warming, has actually increased the amount of abundance of cases. So a little bit of a pitch here. And so malaria, very complex, and I'm not a parasitologist, so Rob taught me everything I know. Yes? Yeah.
Do that in a mouse model. Nice animal model. Either subcutaneously or IV, and compare and see, metastasize.
I give you half a gram. We need money to do mouse studies. So I'm a chemist, right? My funds are usually by NSF, sometimes NIH. We're really, really unfortunate with that. Nobody gives you money to screen, like going around the globe, which is a fun part, and finding the microbes. Once you have an activity, that's where you can write a grant for application. Nobody has pursued mensacarsin for the cancer area. We didn't get the funds. We did some ADME studies, like, is it actually any good in a bloodstream? And this looks all fantastic. It's a very stable molecule, actually. So I would love to collaborate to, we talked about hollow fiber assays and things. How can we bring that more into relevance? Yeah, absolutely. I still think there's hope because there are many metastasizing cancers that need stopping in a way. And it does limit, the side effects are hopefully, as we see from in vitro work, not as bad because most cells get affected by metabolism, but they're not dying. We don't see any other markers of apoptosis, autophagy, anything of cell death you can think. So yeah, we'll love to do that. That's why I'm pitching all these molecules for you guys and be like, I like this one the best. So we do not see any apoptosis and other cell lines than melanoma. That's been tested. We had like five cell lines from the 60 cell line panel. We don't see any signs of apoptosis there, but that's all just, Western blot kind of work. It needs to be more thorough for sure. But hey, for chemists, we're pretty proud, yeah. But no, it should go. I mean, I would love to put it in mice, but what I'm hearing is a mouse is $1,000 per pop, right? And someone has to fund us first to get that, right? Is it not? Okay, we're talking. Okay, fantastic. Okay, so Plasmodium, we had some hits here, and what the initial study was done by Sergio Wittling and Mael Duffy, and they're both part of the Medicines for Malaria venture. And so what they did is they first made me study malaria because it's a complex life cycle. I have never worked with parasites in my life before. Still like horrified and pretty scared. But this is just an overview basically that what I learned is you basically have a life cycle in the blood stage, you have a liver stage, and they're different phenotypes. So you have to make sure your malaria drug is hitting either both of those stages, that's the best case, or where does it hit? Does it hit the schizons? Does it hit in the bloodstream? You want to make sure you eradicate the whole life cycle of a parasite. If I probably get a bad grade here. All right, so, okay. Malaria has drugs, and if some of you might have had a malaria prophylaxis before, it's not a good treatment. So sometimes you take this before you go to a country with malaria. So here are some of the old ones, the chloroquine, artesunate, atovaquone, permethamine. So these are all molecules that work well, but they have lots of side effects, right? If you take that as a prophylaxis, huge side effects, very unpleasant. I also put in roughly the nanomolar activity that they're having. So that's the efficacy on the parasite in different stages. And then I put mensacarcine here. So they tested mensacarcine. We are not as potent. We are in the 100 nanomolar range, but it works really well in the blood cells. That was the first assay they did. And then also it works at the liver stage really nicely. And here comes the clicker that makes me excited. It's active against all the resistance isolates they have on these drugs up here. So that was really cool. First time I show this, so it's really just a screenshot. So what you see here is the standard Plasmodium parasite, and these are all resistant strains for all of those different drugs, chloroquine, artesunate, atovaquone. We retain nanomolar activity while they lost, sensitivity to these other drugs. That was great. But even more cool, and that's where I jumped out of my chair, is there are always experimental drugs currently in development, right? So here's one from Merck, here's one from Novartis, very different targets in the Plasmodium parasites, but all of them, again, Plasmodium, really fast in making resistance. You make resistant mutants for these new drugs in development drugs. And mensacarcine is still active. So even the one that may be next in the pipeline here, we don't have cross-resistant with them, and we are still killing when the parasite is not sensitive to these new drugs anymore. So that was really, really exciting. So how did it bring us to Rob? Well, it's very hard to get funding for Plasmodium in the US. Maybe I'm just the wrong person writing the grants, but it is just very, very hard because it's not something that affects you and me on a daily basis, right? So when I met Rob, she talked to me about, hey, there are actually other apicomplexer parasites that I never heard of. And she, really gave me background information about Cryptosporidium parvum and how is it an intestinal parasite, how it's very small, living here, intertestal, but not cellular, intercellular. How was that? Really interesting. Exoplasmic. So I learned a lot, and it's a beautiful, it's a parasite that we can study to learn for other parasites, but obviously has also direct applications as causing diarrheal diseases, especially in immunocompromised patients, HIV/AIDS, cancer therapy, you name it, right? And so we looked into what mensacarcine is doing in this system, and that's the first time I was able to show it, thanks to Rob. She and her student, Rob, tested it. I always call him Rob squared in my emails. They tested it. They have a beautiful luciferase outread assay, phenotypic assay, as I understand. And we can see the nanomolar activity is retained here. This is done in HCT cell line as a host cell. And then it doesn't affect the cell lines. So that was a very, very promising in vitro work. And what we've been doing now is we can make this mensacarcine fluorescent probes again to show maybe we can see where it is in the cells. We can pull down maybe with molecular binding profiles. Can we pull down the target? And then obviously the goal would be to go in mice, right? Never thought I would work with crypto, but here we are, and it brought me here. So I'm really happy for that. So thanks, Rob, for that beautiful collaboration and pushing that molecule forward. Yes?
Have you done PKPD study?
We have not all of the ATME studies, and yes, we have to do more. I paid a little money to see stability in gastric fluids, plasma, those initial studies. We haven't done any distribution in the mouse or anything. There is a beautiful, I'm part of pharmacy at the University of Florida, so there's a beautiful testing center, but it's still for fee, right? So we were kind of, The molecule, or sorry? It's decent, right? It's 420 molecular weight. It's right there with attenosate or other molecules. We don't see any activity in any bacteria, which you think about microbiome problems or anything. So it's clearly something that affects eukaryotic cells and these parasites. All right, I have a little time. Maybe I'll make the next stop in our little journey. Yes, we go there. We go first to the Arctic, though. Let me go back. Sorry, I want to go back. So next stop, we have 2 stops left. One is the Arctic Ocean. We were able to collaborate with people at the University of Tromsø. And if you look up at the map, that's the northernmost university in the world. So it's a very fun place, all the way up here. That's where they're located. And they go on frequent cruises, explorations into Svalbard, which is this archipelago here. In German, it's called Spitzbergen, so I knew that from lots of reading before. And so a long time ago, they went and got samples from us, and samples are water samples and sediment samples. And they're usually like methane seeps. So there's areas where methane is released, really interesting floor geochemistry that we got samples from. And the graduate student involved is Majorly Aaron Marshall, now, and then before was Zoe and Annabelle. And so here, that's how it looks like. We get like pores, we get like grappers from these samples. We take the effort to bring out the bacteria. Luckily, less bacteria and a little bit of dirt from the ocean than it is from your terrestrial sources, but lots of bacteria nevertheless. And one thing is we always try to find new applications, right? I think you saw that in the seminar. But one thing was always bothering me. We need new drugs for pain. And that's a little curveball here. So everybody was asking for pain medication. Opioid crisis is everywhere, right? Where can we find non-addictive compounds? And natural products is hard to study, right? How do you study natural products or the extracts, really, the extracts, right, for pain activity? But it was a challenge we accepted. And we worked with James Strother at the University of Florida and before Oregon State, so it was great. It's a long time in the making. And what he does is he is a zebrafish neuroscientist, and he takes very young zebrafish larvae, 3 days post-fertilization. Oh no, hatching, I guess, not fertilization. Oh, no. So little tiny zebrafish larvae. And the cool thing about these larvae is they can't swim yet. They don't have an inflated swim bladder. So they're just stasic. They're just as little sticks. They look like that, really. They just align in this well. They don't move at all. They just sit there. But the cool thing is they have a fully developed receptor system. So if you give them wasabi or mustard, they start twitching. They don't like it, right? So they have a fully evolved, for example, TRPA1 receptor mechanism, which we can use mustard oil to give them the twitch, right? So something that is really annoying them. And so, hey, that's kind of pain, right? Because a TRPA1 receptor, that's your peripheral neurons for chronic pain involvement, right? So if we can stop this, if you take topical patches with menthol, this can work because it does connect then to your CNS, your central nervous system. So he got this assay going using this larvae. They're not moving if you don't give them an obnoxious compound, and it's fully automated. So the longest it takes is actually to put the larvae in the well plate and for us to put our chemistry in there. The rest is automated. He just plugs it into his system. Even the analysis of movement is fully automated. So great programming there. Why zebrafish? I don't know if I have to pitch this here from you guys, but zebrafish is actually great. Sometimes I would say boldly superior than mice, just saying that, because many of the compounds retain efficacy in the zebrafish. We have a lot of disease orthologs. And in vivo testing, right, we are not just taking a cell line again, we're having the whole animal, the whole metabolism. So ADME study is basically built into our system. And so what we're doing looks like that, and it's a little complicated, so I'll walk you through here. So here are 6 zebrafish larvae. Each of these rows is 1 larval zebrafish, and there's 6 in one treatment. If it becomes golden, they're moving. If it's black, they're not moving. And what we're doing is we're giving them our chemistry, our microbial juices, and pre-treat these zebrafish with our chemistry. We wait a while, because sometimes they don't like it, as you see here, like, ooh, what kind of juice is that? But then we give them mustard oil. We give them the twitch, right? We cause pain. So we give them this twitch here, and then normally in the standards, they have to move, right? That's the activation of the TRPA1 channel. but maybe we find things in our extracts that are analgesics. So if we have an analgesic, you see something here, they're not moving. So it's a really cool assay. And it does work. We've shown it now. So this is our library. We have probably 8,000 bacteria and fungi in cultivation. We make those extracts. And what we did is we tested that assay, robustness of the assay. Is it high throughput? Is it medium throughput? Does it give us good hits? And it's pretty exciting. So we do see extract that kill. Many things are toxic. We do sometimes see things that reduce pain. That's what we want. And so here are just the stats for the assay. But long story short, here's an example how it works. So we take our extract, for example, this one here, it's a fungal extract, and we see, yes, the zebrafish doesn't like being exposed to these extracts, but hey, after a while, they're kind of okay with that, kind of chill, right, back to baseline. And then we give them wasabi, right? And the control is not liking the wasabi, but our extract has some analgesic magic. So now we need to figure out what is the analgesic in here. And so we do something in natural products. Again, I told you, 1,000 metabolites, lots of compounds. We need to narrow it down. And so that's what we did here. We did a behavior-guided fractionation. It's a funny word. What we did is we fractionate our extract, right? Make it less complex and test and test and test. And let the zebrafish guide us to what's active. And what you see here is, well, there are some fractions that abolish the pain response, totally abolish it, and some retain it. So here's probably our compound of interest. And we can go down and down the rabbit hole until we have a small molecule that's pure and test it, and that's what we got. One of our fungal natural products down here, chalaniline A, this is a molecule, has a really nice dose response in the pain here. So at high concentration, no pain at all, lower concentration, you know, a little bit of pain. So we have something that can find us new analgesics in mixtures. That's important because we have 8,000 bacteria. How do I tell the student which one to pick, right? We need to make a quick extract and prioritize. Next step is, and it's good to have a neuroscience in-house, he does 2-photon calcium imaging on the brain. So what is that? It's basically he takes a larval or adult zebrafish, puts it in this agarose bed. The tail can actually move a little bit. The head is kind of immobilized, and it's a living fish. And it expresses calcium indicators that fluoresce if a neuron is active and firing. So you can see a zebrafish think, act, neurons firing in real time in a live fish, right? And so that's what we're doing. We're putting on our little candy here. That's our zebrafish brain to orientate you to the right. This is the eyes. I never know where top and head is. This is the eyes. Here's the tail area. And if I play this, this is a zebrafish without any treatment. So this is a zebrafish being immobilized in this agar, just thinking, doing its basal activities in the brain. Maybe sometimes freaks out a little. I can't move really well, what's happening, but you know, it's there, it just thinks. At 20 seconds around, we are adding the AITC, the mustard oil, the one that is actually inducing our pain response. And here in the bag, it's called the area postremer, I was told from our neuroscientist. That's where the TRPA1 receptors are. That's where it senses wasabi, mustard, and all the nuisance coming into your system, right? And the whole thing is freaking out, right? All the neurons are like, what's happening? Get me out of here. So that's cool. We see this with our mustard oil. Next thing is, how does it do with our fungal natural product? And I don't have the video yet. He's owing me one there. But what we see in our fungal natural product is actually same area. Our natural product, I showed you, is also affecting the area postremer. I'm sorry, I switched the zebrafish brain on you. But here, this area is where it acts, similar to our allyl isothiocyanate, the mustard oil. So we have a TRPA1 agonist, very likely. We also have some responses here. And I was wondering, what is that? And he's like, that's the olfactory system. Your stuff stinks. I'm like, okay, got to work with that, right? But we have some in vitro data expressing the cell receptors in cells, in HEX cells. We have a TRPA1, this is the population here, TRPA1 agonist found from our mixtures. And now we were lucky to work with a synthetic chemist, Paul Blakemore, really talented group of people, and we can make this molecule now in more amounts, always important. And now we can test it actually more, what population in the brain. And so, because I'm running out of time, I want to just show you she started with the Arctic Ocean, right? There was something, yes. We used this assay to screen our Arctic Ocean bacteria, and that's Erin, who just defended in March. She's now Dr. Erin Marshall. And she took this mixture of Arctic Ocean bacteria, lots of bacteria, got their extracts and tested them in this assay. And really quickly we see, oh, there's some that are not as harmed. There's some analgesic maybe here that she can try to find. And she did. That was great. So she moves forward in some assays, and this is a lot of bars, but what you can see is there are some areas here where the fish are having less movement. That's good. We don't want movement. We want analgesics, right? So, and there are some that are not good. So no stimulus response means dead fish. That's not good. But we have some where the movement is lower, the pain is lower, and the fishies are happy. And she can see this right there. And these are compounds, all unpublished results coming up in the next few months. Very exciting. Small, simple molecules. These are called benzotiazines and 1-benzothiazole. I have never seen so many with sulfur before. So it's from the marine ocean, deep Crocobacter strain from the Arctic Ocean bottom. And they are analgesics. So we have now the data, oops, go forward, here in our assays where we can test them. It's a higher dose we need in the zebrafish, but this is the most active compound that is producing the response in our extracts. So she's writing this up. It's a cool bacterium too. There are only 1 genome published, only 3 strains published on in the world. No natural products, so we are new to that. And there's more to it. And actually the skeleton here gave us ideas. We had a summer student, and I had to write this down. You don't have to read this, but we had a summer student really enforcing international collaboration. This is a Pakistani student that came during her PhD time to me. Took us a year to get her visa because Pakistan is not on the wish list for many students to come. But when she came, she was only here for 3 months. She made 28 of these compounds, and 2 are currently in patent with UF. They have the core. I can show the core. They don't have some other features to make it a selective analgesic. So very important, and so happy that she can come after all. Mom of 2 had to come 3 months alone. They denied the visa for the parent family. So lastly, I promised Rob, we were invited to go to an expedition to Antarctica in January. Sounds lots of fun. I didn't go. I am not good on boats. I'm not good on planes. I'm not good in the cold, sorry. So I sent Federica, a postdoc at the time, and she actually, we made overnight a little flag she could bring. And then she came back with all these samples. And this is all facilitated by our Chilean collaborator, great guy, Jaime, and she had a great time. So we have, I think, over 100 bacteria from there now. They've all been in the pain assay. You know, no pain, no gain. And we also look into other activities. So there might be new parasitics in there as well. So just give a shout out that this was an amazing opportunity. Great to work with people in Chile. And that's it. I'll just stop here right now. We have a great team, a small team. This is our back view at our beach day. Again, here's Aisha, summer student, Erin, who graduated just now, postdocs, undergrad who's now at Berkeley, unfortunately. Good for him, bad for me. And then new graduate students all coming in. Funding, obviously, nothing is possible without current funding. And again, NIH, we need the impact. We need to bring the molecules to NIH. And I appreciate you all. Thank you.
And you're cut off from questions because we're out of time because you had too many questions during the seminar. So we'll take one from Dr. Geddes here.
Hi, thank you for coming, visiting us, and sharing all your work. So my name is Alonso. I'm a veterinary anesthesiologist, and I work on pain as well. So I'm really intrigued by your model. I was just wondering, how do you differentiate between a sensory effect versus a motor effect on your assay?
Yeah.
Repeat the question for Zoom, just in case it's not working.
I barely understood the question, but I get it. How do we distinguish the compound's pain activity in terms of modulatory, like desensitizing, if I got that right, or what kind of? Yes. I got it. Okay. That is a great question, and my neuroscience collaborator, Jim is on that. I'm so sorry that I'm not a good neuroscientist in that regard. So, but he looked into that, and one thing is, so the brain imaging gave us some ideas because the population of neurons in the zebrafish is really well mapped. He actually printed out a map for me saying, you know, that's where the neurons are and the different types and how they're then interlinked, obviously, and transverse in a CNS. So he has some ideas there. There have been work done where they ablate certain connections, certain connectivities, and then see if the effect is still remaining. We are right now more concerned. We see desensitizing in this molecule, again, similar to AITC, similar when you eat wasabi or capsaicin, right? You can eat more, you desensitize. That's the same we see with this molecule too. So we need to see where does it come from? Is it deactivating of the receptor? Is it covalent binding to the receptor? But I'm looking forward to pick your brain more because we just published, so the assay for the screen is published now by my colleague. I'm just happy to go on the right. I think it's on the link on the bottom. And he did due diligence, really. He tested, this is a paper that just came out 4 weeks ago. He did due diligence. We did all kind of controls. So from different morphines, different analgesics, benzodiazepines, everything you could find in a pharmacy. We grabbed, if we were allowed to use it on the fish by IOCAC, but we grabbed all of them to due diligence and see what can this assay actually detect and what not. And that's all in that paper, and he would be the expert to us. I also want to point out that these are actually first authors undergrad, second year grad student, undergrad. So amazing work by just a small team, but I might catch you. I'd like to hear more. Thank you, and learn more. Appreciate it. Thank you, interest. Yeah.
Oh. We have one more question.
Thank you. Yeah. So Sandra, I really enjoy your talks, I like the story a lot. I'm not going to ask about science, a part of it. I just want to ask a general question with regarding to the status of natural product research these days. My impression is, I mean, lots of pharmaceutical companies cutting those things off from their research and development. So is there any issue with the natural research product research these days?
It's a very good point, and yes, so
Sorry, could you repeat the question?
yes, question is, natural product research has been kind of downsized in industry for sure, and is it an issue with the source of these chemicals? I think that the answer is twofold. One is, yes, many industries, large companies have downsized looking at natural products because it's complex. As you see, once we find a hidden extract, you have to have the manpower, woman power, student power to get it down to the molecules. It's very hard to approve mixtures, right? It's working in some botanicals, but not usually in our sources. So finding the pure molecule is usually pushed nowadays to academics, find the molecule, and many, many startups. So that's actually the second part of the answer is, while it's not maybe at, antimicrobial research shrunk, natural product research shrunk, and all of the big companies, it does emerge more in startups. So there's LifeMine, there's Hexagon, one just does fungi, one just does bacteria, and they do obviously genomic information because all these molecules are genetically encoded, right? If you know what you look for, you can find these biosynthetic machineries that make them, and nowadays the promise is, I don't need the fungus or bacterium to do this anymore. I take a heterologous host, right? And you might be independent, getting better yields. So it moved from the big companies to the small companies, but they are abundant. And the good thing I must say for my students is all my students, many of them don't do postdocs, they go straight to industry. Because the skill set of isolating a chemical from any mixture you give them and figure out the structure, the three-dimensionality of the structure is a skill set that's still wanted. Actually, there was last week a big shake in the LinkedIn and the startup companies, and 2 of my students lost jobs, previous students, one postdoc, one student, but one found it within a week, a new job, the other one is looking, she's a little bit changing and branching out. So it moved into more startup space, and it used to be more into genetic information, like the random searching that I'm doing, 8,000 bacteria, 3 students, what's the odds, right? You have to have smart assays to screen, that's where I always try to justify my approach. We have assays that are unique enough that gave us hits in the past. But having the genetic information is much more powerful. So yeah, it moved from big companies to small. Luckily, the skill set that my students learn is wanted. Good, right? No AI can solve a structured 3-dimensionality. No AI can work on a milligram or 2 usually. So that's good. Are we missing out? Yes. Because as you all know, biodiversity globally is shrinking. We haven't even, bacteria and fungi, we haven't even touched what's all there. And, if I speak to microbiologists, we can only culture maybe 5% of the microbiology diversity that's out there. 95 is this dark matter of microbiology that we can't even assess. We can't culture. Everything in my lab has to grow in a little agar plate and be happy, right? 95% of things are not growing on an agar plate, will never be screened, right? And the genetic information is there, so people do metagenomics now, finding all the genes in one little ecosystem and saying, I can see natural products here. I see a ton of natural products, right? How do we get them? How to express them? What's their function in ecology? So we are missing out, and the shrinking biodiversity is not helping. So it's a big pitch for us to say, save and secure those areas, microbes, plants, all of them, because we haven't even studied them as much as we should. Do natural products still inspire compounds? Yes, because you see startups that have good structures with some kind of interesting activity, they get sucked up and purchased. Cubist was one, new antibiotic come out. So they still inspire drugs. They evolve for something, right? Why would a microbe and bacterium or plant spend so much time making this molecule, right? To affect some receptor, some DNA, some protein in their own mechanism or outside in their niche. So this evolutionary development we can't ignore. It would be just stupid, right? And you see structures. So my postdoc just found a complete new carbon skeleton. It's mind-blowing. We still find these things. Even the organic chemists, don't tell them, but they put all the carbons together. We know how to do that. They have never seen that structure we have on my desk right now. And that's so cool, right? So there's so much diversity and structural diversity that we can't mimic by AI or organic chemists. So it's a concern. But I think modern technology might help with that. And genomics did definitely help with that. Knowing where natural products are and then maybe getting them without the microbe and cultivation. Hope that's the future. Tough one.
Okay. Thank you. We have the research office has some Minnesota gifts for you. If anybody has more questions, there is some spots with the student / postdoc lunch in 495 upstairs, and you can join them. Thank you.